Hello, everyone, and welcome to today's webcast. I'm Cheryl Rogers, the Director of Marketing here at Golden Helix. It's my pleasure to introduce Hilal Al Shekaili, a PhD student at the University of British Columbia in the Department of Medical Genetics. Hilal was our second prize winner in our annual abstract competition this past winter, and today he will present his research discovering novel candidate genes for pyridoxine-responsive epilepsy. Hilal, I will give you the honors.
Thank you, Cheryl. I'm pleased to welcome you all attendees. And the title of my research project is, as you can see on the screen, Genetic Basis of Pyridoxine-Responsive Neonatal Epilepsy in Consanguineous Families. This is an outline of my talk, so I'll give an introduction about the absorption and metabolism of pyridoxine in the human body. And then I'll go through some of the knowledge gaps on pyridoxine metabolism and transportation. And then I'll speak about the disease we are studying, which is pyridoxine-dependent epilepsy. I'll speak also about the phenotype of the studied family. And then I'll go through the steps which we did through in our molecular work, and then our results and the next steps. Although pyridoxine is a word that is used interchangeably with vitamin B6, actually vitamin B6 is a generic term that refers to 6 interconvertible pyridine compounds, or they are called also vitamin B6 vitamers. The structure of the compounds are on the figure shown on the slide. There are 3 basic compounds. It is the pyridoxine, which is an alcohol, and the pyridoxamine, which has an amino group, and the pyridoxal, which has an aldehyde group. The other 3 compounds are simply the phosphorylated compounds of the 3 basic compounds. So we have the pyridoxine 5' phosphate, pyridoxamine 5' phosphate, and pyridoxal 5' phosphate. The most important compound out of these 6 vitamers is the pyridoxal 5' phosphate, simply because it is a cofactor for over 140 enzymes in the human body. The major classes of these enzymes are oxidoreductases, transferases, hydrolases, lyases, and isomerases. The figure in the circular shape shows the structure of pyridoxal phosphate in the middle here, and then it lists all the EC or enzyme commission numbers for the enzymes that are dependent on PLB. And as the larger category of enzymes that is dependent on PLB is the aminotransferases. All living organisms rely on vitamin B6 for their existence. However, only microorganisms are able to make vitamin B6 de novo. All other organisms, including humans, need to acquire vitamin B6 from diet, and then inside their bodies they can interconvert different vitamers of B6 by use of the salvage pathway. The figure here shows the de novo pathways. There are 2, so far there are 2 de novo pathways for the synthesis of vitamin B6 that are discovered in other species. The first one is called the DXP-dependent pathway, which is found in some eubacteria. The other pathway is called which is shown here, is called the DXP-independent pathway for de novo synthesis of vitamin B6, and this pathway is found in, it is more predominant compared to the DXP-dependent pathway, and it is found in archaea, most eubacteria, fungi, and plants. The DXP compound simply refers to the compound here because this compound is missing from the pathway, the second pathway. This is the reason why it is called DXP independent. The human and animals rely on another pathway, which is called the salvage pathway, which is simply the interconversion of the different vitamers of B6. So they acquire the vitamers from the from diet, which is coming from the de novo synthesis from other species, and then they interconvert the different vitamers using the salvage pathway. The key enzymes in this pathway is the pyridoxal kinase, which is BLK, BMB oxidase, or pyridoxine amine phosphate oxidase, and a number of phosphatases. So now I will go through the steps or the hypothesized route for absorption of different vitamins of B6 in the human body. So as I already mentioned, humans obtain vitamin B6 mainly from diet, and also there is some absorption also occurs from bacterial sources, which is the normal microflora dwelling in the large intestine. So the figure here shows the route by which the different vitamins are absorbed by the human body. So phosphorylated vitamins, or the ingested phosphorylated vitamins, are first dephosphorylated before absorption. So the phosphate group is removed by the action of intestinal phosphatases. And we have a vitamin that is called pyridoxine glucoside, which is mainly coming from the plant. So it is a pyridoxine with glucose molecules attached to it. And for pyridoxine glucosidase, the glucose molecules need to be removed prior to absorption, and this is done by the action of also intestinal glucosidases. So the dephosphorylated enzymes, or the dephosphorylated vitamins reach the blood, and then they are mainly they are cleared mainly by uptake into liver, and inside hepatocytes they are again phosphorylated by the action of pyridoxal kinase into their phosphorylated counterparts, so pyridoxal phosphate, pyridoxine amine phosphate, and pyridoxal phosphate. And inside the hepatocytes, these all the vitamins are ultimately converted into the active cofactor, which is pyridoxal phosphate, by the action of the salvage pathway central enzyme, which is BMB oxidase. Then the pyridoxal phosphate circulating in blood, distributed to all tissues of the body, but before also being absorbed into cells, the active cofactor need to be dephosphorylated, and this is done by the tissue non-specific alkaline phosphate, which is an ectoenzyme. And once the active cofactor, or once the pyridoxal is absorbed into the cells, including cells of the brain, they are or it is again phosphorylated by the action of pyridoxal kinase into the final and active form, which is pyridoxal phosphate. Now, there are a number of missing pieces of knowledge in pyridoxine metabolism and transportation, especially in humans and mammalian species. And probably the most important missing piece of knowledge is the regulation of the salvage pathway. So little is known about the regulation of salvage pathway, and which is the main function of the salvage pathway is the interconversion of the different vitamins into the active cofactor, which is BLB. So BLB is a very reactive aldehyde, and the most prominent example is the disease that I will mention subsequently, about the high reactivity of BLB, and so high levels of BLB lead to disease like sensory neuropathies, and also deficiency of BLB leads to different pathologies, including the main disease of the talk today, which is pyridoxine-dependent epilepsy. This means that BLB levels, intracellular levels of BLB, are tightly regulated within certain limits. However, it is not completely understood how these levels of BLB are controlled within the cells. And it also remains to be resolved how BLB is supplied to its dependent enzymes inside the cell. So, given the high reactivity of BLB, it has to be there should be some special mechanism for BLB to be delivered safely and securely to its dependent enzyme. BLB is a highly reactive aldehyde, as I said, and it can form aldimines with amino groups on other non-dependent, non-B6 dependent enzymes, as well as other amino acids. Moreover, there are a variety of complex compounds which are derivatives of pyridoxine that were detected in the body fluids of humans and other mammalian species. However, the precise function of these compounds is still unknown. And the last gap in the knowledge of pyridoxine is the observation of that 20% of the non-BLB dependent enzymes which are encoded by the human genome have unknown catalytic activity. Also, there is a large gap in our knowledge of pyridoxine transportation, especially in the human body and mammalian species. So previous studies have shown that absorption of B6 through the plasma membrane or transportation of B6 through the plasma membrane occurs by a carrier-mediated mechanism. So there is a special transporter for B6, and it has been shown using studies on the intestinal colonic as well as renal cells that the absorption of B6 does not occur by simple diffusion or passive diffusion, but it has a carrier-mediated way of translocation through the plasma membrane. However, in humans, despite this experimental evidence, so far in humans, the molecular identity of this transporter at the genetic and protein level has not been resolved. Generally speaking, in other species, the only identified B6 transporters are the two transporters in the yeast. And also recently, the transporter BOB1 or burin permease was discovered in Arabidopsis species of plants, which is the first one to be discovered in plants by Silidowski and colleagues in 2013. These people who have discovered the transporter in plants were able to do so by working on mutant phenotypes of plants. So they knocked out the genes responsible for de novo synthesis of vitamin B6 to make these plants are unable to synthesize vitamin B6 de novo. So the only way that they can survive is by absorption of pyridoxine from outside. And when these plants, the mutant plants, showed that they were able to absorb pyridoxine from outside, which implied that there is an uptake system in these plants. And through subsequent experimental work, these people were able to identify a high affinity transporter for pyridoxine in plant species, which is the Arabidopsis. This is a KIG pathway showing the vitamin digestion and absorption in the human body. The in the upper side shows the water-soluble vitamins, and the lower side shows the fat-soluble vitamins. As you can see here, most of the absorption routes are well described in most of the water-soluble vitamins, and the transporters, which are highlighted in green, found in the brush for water-soluble vitamins. However, as you can see here, the vitamin B6 seems to be the only one with the missing absorption route here. The only described step here is the conversion of the vitamins from the phosphorylated forms into the dephosphorylated forms before absorption. So the people who have identified the transporter implants, as I said, accomplished so by working on mutant phenotypes, and also one way of identifying or filling the gaps in our knowledge of pyridoxine metabolism and transportation is by studying also some mutant phenotypes in humans, which are related to pyridoxine metabolism. And one of the well-known diseases in humans which are related to pyridoxine is pyridoxine-dependent epilepsy. This disease is a rare autosomal recessive disease, and the estimated incidence ranges from 1 in 20,000 to 1 in 600,000. The pyridoxine-dependent epilepsy is characterized by frequent seizures in the perinatal period, so it can occur in the prenatal, neonatal, or postnatal periods. And what is special about this type of seizures is that they are refractory to conventional anti-epileptic drugs. However, they are well controlled by the administration of pyridoxine of vitamin B6. And if pyridoxine is stopped, these seizures relapse very soon. This is the reason why the disease is called pyridoxine dependent. So it is a lifetime dependence on pyridoxine. Discovery of the genetic cause of pyridoxine have went through some milestones, and the first case report of PDE was in the 1950s by Hunt and colleagues, and it wasn't until late 1990s when the researchers were able to map the location of, or the locus of, the gene responsible for pyridoxine dependent epilepsy, and it remained the identification of the gene itself remained elusive until 2006, where Mills and colleagues were able to find the gene that is causing PDE, and the gene that is causing the disease is called antiquintin gene, and it is an aldehyde dehydrogenase. It belongs to the family of enzymes called aldehyde dehydrogenases. And the pathophysiology of PDE is simplified in this diagram here. So mutations in this gene leads to inactivation of its encoded enzymes, which is alpha amino adipic semi-aldehyde dehydrogenase. This enzyme functions within the lysine catabolism pathway in the brain, and it is more clearly illustrated here in the figure shown on the slide. So this figure here shows the lysine catabolism pathway, a snapshot of the lysine catabolism pathway in the brain. Actually, this is one type of the lysine catabolism pathways. There are two described in humans: the bibucolic acid pathway, which is mainly active in brain, and the one which is shown here. And the other pathway is called the saccharobin pathway, which is mainly active in liver and other organs. So the mutation in the gene leads to inactivation of the enzyme here, which catalyzes the conversion of this step here, conversion of the bibutyric acid into amino adipate, and blockade of this step leads to build up of the metabolites that are found at this step here. These metabolites are the alpha amino adipic semialdehyde compound shown here, and which is in this compound is in equilibrium with another compound called bipyridine 6-carboxylate, or B6C. So the compound, the B6C, cross-reacts with pyridoxal 5' phosphates. So because of the high reactivity of the aldehyde group in BLB, it reacts with the activated methylene group in B6C, forming, or by chemical condensation, they together form a complex compound which is biologically inactive, and this renders BLB also inactive and subsequently depletes BLB levels within cells. it is a complicated form of this type of pathophysiology, but these people were able to pinpoint the right mechanism, and they were also able to show it in vitro by using the experiment in which they first incubate BLB alone, and using HPLC they showed that the peak of BLB here, and then they mixed BLB with the B6C compound, and after some time the solution was HPLC screened, and the readout showed a collapse of the BLB levels and the appearance of novel complex compounds which are made from the chemical condensation between BLB and B6C. So the discovery of the genetic cause and also previous case reports that showed the elevation of some toxic compounds in the patients of PDE have led to also to the discovery of some helpful biomarkers in for the diagnosis of PDE. So the traditional way of diagnosis of PDE diagnosis was based on the pyridoxine withdrawal. So simply pyridoxine is stopped, and if the seizures recur, then it is an indication of PDE. But because this method was sort of an invasive one, and the discovery of the biomarkers was really helpful for clinicians and also for patients. Because now screening these biomarkers can be done easily using biochemical ways, and elevation of these compounds, mainly alpha ASA in plasma or urine, and also bibutyric acid in plasma, compared to age-matched controls, is diagnostic of PDE. Now, how BLB leads to the development of seizures is explained. One hypothesis that explains the pathogenesis of seizures is the one that is shown here. So BLB is a cofactor for an important enzyme in the brain called glutamic acid decarboxylase, which is responsible for the conversion of glutamic acid, the main excitatory neurotransmitter in the brain, to the main inhibitory neurotransmitter, which is gamma aminobutyric acid, or GABA. So blockade of this step, or depletion of BLB, leads to inactivity of the enzyme that is dependent of BLB, and this subsequently leads to the diminished concentrations of GABA, which is the inhibitory neurotransmitter. This is one hypothesis that explains the development of seizures in these children. So far, the antiquitine gene is the only one that is described to be causative of pyridoxine-dependent epilepsy. However, whole-exome studies and also single-gene testing of antiquitine in other families have revealed that locus heterogeneity exists, and these families were negative for the diagnostic biomarkers and also for genetic testing of antiquitine gene. And it has been shown also that nearly 5% of children who have a clinical pictures that is typical of PDE have no detectable mutations in the antiquitine gene. So identifying this causative gene in such families will help to improve treatments at the first place, and also it will help to fill the knowledge gaps about the pyridoxine metabolism and transportation in the human body. So giving this important goals, the aim of the project, the aim of our project was to characterize the genetic defect underlying PDE in a consanguineous Omani Arab family with 2 affected children who have a PDE-like clinical picture but negative antiquitine biomarkers. Because these families were recruited from Oman, I'll give you a brief idea about this country, which is located in the eastern tip of the Arabian Peninsula, and it has a total population of about 3 million people. The Omani population has a high consanguinity level, about 55% according to one of the studies. And because of the high consanguinity level, they have also a wide prevalence of autosomal recessive diseases. This is the family, or this is the pedigree of the recruited families, and this is the proband. As you can see here, the proband has an also affected sister, so there are 2 affected children in this family, and from the pedigree you can see that the family is segregating other phenotypes. Some of them are really pretty complex, like the phenotype in the double first cousin here, and an Usher-like phenotype in some of the members also. But because we are targeting these 2 affected children because they had a pure picture of pyridoxine-responsive epilepsy. And I'll mention some information about the phenotype of these 2 children. So the seizures started early at the 3rd to 4th week after birth. They were breakthrough seizures with febrile illness, and they were refractory to multiple anti-epileptics. However, they were dramatically abolished with the vitamin B6 treatment. EEG pattern showed burst suppression, and screening of antiquitine biomarkers showed negative urinary alpha ASA, and also the plasma bibutyric acid levels were within normal range. This was indicative of some other cause in this children, and the flow chart here shows broadly the steps that we adopted to investigate the disease in these children. So the first step was the screening of the antiquitine biomarkers, followed by BMBO sequencing, then SNP microarray, and finally whole exome sequencing. More details on these steps. So the screening of the biomarkers was done at the original clinic in Oman, and they were both negative. BMBO sequencing, BMBO is another gene that causes also a pyridoxine-responsive epilepsy, known as BMBO deficiency. The two types of diseases, I mean the pyridoxine-dependent epilepsy and the BMBO deficiency, used to be distinguished by a response to treatment. So PDE is responsive mainly to pyridoxine, while BMBO deficiency is responsive mainly to BLB. However, there has been some recent case reports which indicated that also there is a subcategory of BMBO deficient patients who respond mainly to pyridoxine. This has made some mix-up between the two phenotypes, and that's why we decided to exclude BMBO at first, and so we sequenced BMBO by Sanger sequencing, and it was negative for any mutation. And then we went into the SNP microarray and whole exome sequencing. These two steps will be explained in more details. The SNP microarray was performed by using the Illumina Human Omni 5 quad array chip, and it was performed on the father and the two affected children. The call rates for the samples were pretty high, approaching 100%, and this yielded a coverage of about 4.3 million SNPs. And the SNP density, as calculated by the SVS software, is 1 SNP per about 700 kilobase, with, which gives an information about the high density of this chip. Our aim of this SNP genotyping was to do a runs of homozygosity mapping, and so the data set from this SNP microarray was used to map the runs of homozygosity in this family, and this was done by using the SNP and variation suite SVS software from Golden Helix, and with the following criteria. The minimum ROH was set to 500 kb, and also the minimum number of SNPs per ROH was set to 25, and we allowed the inclusion of up to 1 heterozygous call, and also we allowed the inclusion of 5 missing genotypes in a single ROH, and the maximum gap between SNPs was set to in any ROH was set to 100 kb. So based on this confounding criteria, the primary SVS run yielded about 327 ROHs in the autosomes of these 3 individuals, which are the father and the 2 affected children. But because we have 2 affected children, we have only targeted the shared ROH between these 2 affected children, and so the number was has collapsed down to 46 regions. The figure here is taken from Genome Browse, and the blue tracks here shows the extent of the runs of homozygosity in the affected cell proband and the father. So the largest run of homozygosity was discovered in the affected SIP, and it spanned about 12 MB from chromosome 6, and it included over 15,000 SNPs. The second largest ROH, or this one, the first one was the largest ROH overall, but this one here is the largest ROH that is shared between the 2 affected SIPs, and this one spanned about 9.7 megabase pairs of chromosome 11, and it included over 13,000 SNPs. The second largest shared ROH was found in chromosome 16, and it included about over 10,000 SNPs. Now, using the same SNP array dataset, we performed the copy number variant analysis using the copy number analysis method in SVS, and there was no pathogenic CMV detected using this method of analysis. And I'll just highlight one of the discovered CMV in these patients, and as well as the father. So the plot here shows it is a genome browse image that shows a plot of the log R ratios. And as you can see here, the drop here in the intensity shows a deletion that is discovered in the index affected SIB as well as the father. And this was discovered in chromosome 8. However, by plotting the public database tracks, like the DGV variants and the 1kg CMV track, the discovered deletion or CMV actually was already reported in the public databases, and it has been reported in ClinVar to be likely benign. And another evidence that it is not pathogenic is the fact that it is found at a high frequency in the healthy population. As you can see here, from the genome browse console, you can get an information about the frequency of any of these CMVs by clicking on one of them. So this one shows that the frequency of this CMV, as reported in the 1kg database, has a frequency reaching of 40% in the common population, which indicates that this is a polymorphic CMV. Now, the whole exome sequencing was performed on the mother and affected SIB. Simply because we had run out of genetic material for the father and the proband, so we sequenced the mother and affected SIB from Berkin-Elmer of USA, and the exome capture was done using the Agilent SureSelect library with 100 base pair per end construction, and using the Illumina HiSeq 2000 for sequencing. This figure shows a flowchart of the whole exome sequencing analysis pipeline, the steps that are implemented in our bioinformatic analysis pipeline of the exome data. So starting with the primary alignment, realignment, and then variant calling, and also these steps followed by about 5 filtering steps. The steps are first filtering out the low Q or low quality variants, and then filtering out the intergenic and interonic variants using the SNP-F program, and then also exclusion of the polymorphic variants, those which have a frequency of higher than 1%. And the fourth filter, which is applied, is the removal of the variants that do not have a direct coding change. And also, we have our own database of whole exome sequencing projects, and we compared our sequencing here to our database, and any variant which is observed more than 10 times in our database was subsequently removed from the final list. The column on the left shows also how the number of variants drop after applying each of the filtering steps here. So as you can see here, we started off with about over than 350,000 variants, and finally we ended up with about 13,000 variants. Now, focusing only because our genetic model here is an autosomal recessive, so we only call the variants which are homozygous recessive. What I mean by homozygous recessive are the variants that are homozygous on the affected child and heterozygous on the mother. So we had about 38 homozygous recessive variants. The overwhelming majority of these variants were single nucleotide substitutions, causing missense changes, and a few of them were indels, and the rest were all affected the splice sites. Now, because we had the runs of homozygosity on this family, we only focused on the variants that are found inside these runs of homozygosity or fell at a close proximity of the boundaries of this runs of homozygosity. And by doing so, we ended up having only 11 variants that had genomic coordinates overlapping with the ROH in this family. Now, to prioritize these 11 variants, we have set our candidate gene hypothesis to comprise any gene that is involved in lysine degradation pathway, because PDE is caused by a defect in this pathway, and also proline metabolism pathway, simply because there is another phenotype which is also pyridoxine-responsive, and it affects the proline metabolism, and also any gene that is involved in vitamin B6 metabolism or transportation, because the B6 transporter in human body as well as mammals is not known yet. We have included any sort of protein that has a transporter activity. So none of these 11 variants had a clear overlap with these or with these metabolic pathways. However, we could find two good candidate genes for this phenotype based on their functional relevance. I'll go briefly through these two candidate genes. The first candidate gene belongs to the solute carrier superfamily. So this is a large superfamily which has an extensive number of genes that are involved in transportation of nutrients and other molecules through the plasma membrane. And this gene, the candidate gene, is expressed everywhere in the body, and it is known to mediate the transportation of the ions through the plasma membrane. It is not well described in literature as well as databases, and the only known function of it is to mediate the movement of certain ions through the plasma membrane. So our interest in this particular gene stems from the observation that three members of this solute carrier superfamily have been already shown to act as vitamin transporters, mainly the SLC19A2 for thiamine, and also two other genes from the same family which are described as transporters for folate or vitamin B9. And also, in addition, the mutations in the gene, the thiamine transporter gene, is known to cause a Mendelian disorder known as thiamine-responsive megaloblastic anemia, and also the similar thing to do with one of the folate transporters, which causes also mutations in this gene also has been reported to cause autosomal recessive hereditary folate malabsorption disease. This is a genome browse snapshot showing the exome data by lab, and showing also the location of the mutation in the first gene. As you can see here, this is the reference human genome sequence, and it shows that it is a C to T conversion, and it is highly covered by over than 100 reads, and the second candidate gene is an enzyme. So the first one is a transporter, and the second is an enzyme that is highly described, or that is highly expressed in the ovary and testes, as well as within discrete areas in the brain, and it encodes a type 2 integral membrane protein. So this enzyme has been described to cleave a neuropeptide, and this neuropeptide is expressed in the central nervous system as well in the periphery, and it was thought to function as a neurotransmitter. The second image here shows also the locus of the second mutation in the second candidate gene, and it shows that it is a G to C conversion, and also it is highly covered by exome reads, over 100 reads covering that mutation. Now, because these are our findings so far, and both of these variants have been Sanger confirmed, and both have segregated in the family, so we couldn't so far resolve the two candidate genes into one. But our next steps will be implemented towards narrowing down these two candidate genes into one by recruiting more families from the original population with the same phenotype, and also by collaboration with international teams working on this phenotype. And we are also open to share our variants with these people, and also an important assay to do is the metabolomics assay of the plasma and urine samples in the of these patients. So metabolomics assay will give us a comprehensive profile of the small molecular weight metabolites in these patients that went into abnormal levels, and by doing so we can pinpoint the defective biochemical pathway, and then the final approach will be the functional validation of the candidate gene. So I will take this opportunity to offer this collaboration with anyone who is working on similar projects. So we will be pleased to recruit such families that have a clinical picture of unexplained neonatal seizures that are irresponsive to the anti-epileptic drugs, but that responded well, and they were well controlled with administration of pyridoxine. And also it has the already known causes for pyridoxine-dependent epilepsy, and generally speaking of pyridoxine-responsive epileptic encephalopathies were excluded, either by biochemical testing or by direct genetic testing of the causative genes, such as antiquating BMBO and ALBL, which is coding for the tissue non-specific alkaline phosphatase. We are also, as I said, we are also open to share our discovered candidate genes with anyone who is working on similar patient cohorts. This is my email for any contact, and also I'm showing the email of the our project lead, Dr. Clara Varna-Kernabik, and she's chairing the International PDE Consortium. And the final slide here shows the acknowledgement of our research team, as well as our collaborative clinician from Oman, Dr. Khalid Al-Dihli. And also, I would like to acknowledge the Golden Helix team for giving me the opportunity to present this webcast, and also for their long-standing help in SVS as well in other technical issues, how to do the different analysis with SVS. And thank you all for listening, and I will be happy to listen to your questions.
Thank you, Hilal. We will take any questions. Feel free to go ahead and enter them in the questions pane in the GoToMeeting interface. I have just a couple of quick announcements before that. The webcast recording and slides will be up on our webcast today or tomorrow at the latest, and I'll email those to each of you, so look for those in your inbox. As I mentioned at the beginning, Hilal was one of our Abstract Challenge winners, and he had the opportunity to present his work today, as well as winning a license of the SVS software. Excuse me. We will have our third annual competition coming up in December, so if you're interested in participating, feel free to register for news from the Golden Helix team. You can do that right on our website. Our next webcast is scheduled for September 23rd, and we will be unveiling our newest feature in our VARSeek software for clinical reporting. Look for invitations to come out tomorrow or Friday. And that's all for me. So we will go ahead and take a couple of quick questions. Let's see, did you look at the ROH regions in unaffected family members to narrow down your candidates?
Yes, I did. So as I said, we took only the shared ROH in the affected children. And for example, if the same region is also found in an unaffected member of the family, it will be unlikely that it is the targeted region. So we did so. We had the SNP data on the father who is unaffected. We have also SNP data on the some of the double first cousins of this family, as well as some of their uncle and other family members. So we put all the run of homozygosity together, and usually the ones that the run of homozygosity, which is found in an affected cell, it is usually uninformative, and it is likely to be not the one that includes our targeted gene.
Have you done any functional analysis for your identified genes?
No, not yet. So as I said, we had two discandidate genes, and they were poorly described, either the transporter or the enzymes. They are poorly described in literature. So before going into functional assays, we thought to narrow them down into one, so that we can invest into the proper gene. And the only functional assay which might be helpful and easier to do is, as I said, the metabolomics assay, which can give you a comprehensive profile of the abnormal metabolites, and it can lead you away into the defective biochemical pathway.
Okay, thank you. That's actually all the questions I see at this time. If there are any more, feel free to email them to us at info@goldenhelix.com, and I will make sure that they get to Hilal, and he will get back to you with answers.
Sure.
Thank you so much for your time today and for presenting your work. And thank you to everyone out there for attending, and we will see you next time.
Okay, thank you all.